Okay, welcome to the first part of our first lecture, a brief history of microbiology. Today we are going to go through the PowerPoint and identify notable microbiology figures, list their accomplishments, compare and contrast prokaryotes and eukaryotes known to cause disease. So we'll go over the six um, types of microorganisms. And then name classification schemes and use bacterial nomenclature appropriately. And there we'll use binomial nomenclature, a system of two names. All right, and then so for this first part of the recording, we'll go over the case study, we'll go over some ancient contributors, and then we'll go over binomial nomenclature and taxonomy. Then I'll end the lecture and we'll go over the prokaryotes and eukaryotes, the six types of microorganisms, and then finally we'll get into the rest of any notable figures we might have missed and list their accomplishments, and as we move into the chemistry right after this. Okay, so I hope you have looked over the case study because we are going to move right into it. Okay, so like I mentioned in my first video, if you open up the lectures before these videos are posted, it'll give you plenty of time to kind of discover the case study and know what we're talking about. So that way, as I go through it now, you kind of can keep up. All right, so Cora, remember, the mother of two, started feeling like she had symptoms that correlated with meningitis. So the doctor ordered a lumbar puncture. The lumbar puncture is a technique where CSF, or cerebrospinal fluid, is extracted from the lumbar vertebrae. Normally, the CSF is clear, like we see in the picture beside here. If it's cloudy, it's usually a sign of a bacterial infection. Cora's CSF was actually clear, so it was clear that she was not. It was clear that she was not under the effects of a bacterial infection, although the doctor prescribed uh, antibiotics nonetheless, um, just to make sure it wasn't a bacterial infection somewhere other than in the CSF region. But we also go through it and we realize that it wasn't our typical disease that she had. In fact, it was a improperly folded protein that was in her CSF instead of bacteria. And so in microbiology, we study infectious agents such as bacteria, fungi, viruses, helminths, which are kind of worms. But yet, Cora's condition wasn't caused by any of these, actually. Instead, it was the prion. A prion is the improperly folded protein. Prions we'll learn about with viruses shortly today in the next video, but also when we go over viruses as a whole, we'll learn prions at the end there, and it's prion. Um, so this led to her diagnosis of Creutzfeldt-Jakob disease, a fatal condition brought on by prions. And unlike the viruses, helminths, fungi, prions, they actually lack genetic material. And so this pushes our boundaries of what microbiology can be. What is microbiology? And now that we're studying prions, what other things will microbiology have to discover in the future? So we can see the vast expanse that microbiology covers and many of the unknowns that we'll still face. And with that, we get to move on to some of our first ancestors and what they knew and the ancient contributions that they made. So before we start, we see that we have three philosophers here, all busts and statues. And why are we studying about them? And it's clear, because they wrote their stuff down. They wrote everything that they needed to down, all their theories, and then throughout the thousands of years, people have copied it again and again and again and carried it with them. And that's why we still study about them. So, if you're ever wondering why we learn about these people, it's because they wrote it down. Alright, so here we have Hippocrates, the father of medicine. When you take the Hippocratic Oath, you are basically taking the oath that he made: do no harm. He laid the groundwork for understanding that diseases could be caused by environmental factors. He believed that health was affected by air, water, and the places that we live. Meanwhile, Thucydides, during the Peloponnesian War between Athens and Sparta, when they were fighting, he documented that the survivors of the so-called Athenian plague at the time did not get sick again when they fought down the road. And he kind of hinted at the concept of immunity. Alright, and then finally we have Varro here, a Roman scholar, wrote his agricultural treatise about tiny creatures invisible to the naked eye. They could enter the body and cause serious disease, and though this sounds remarkably like modern science, he was Roman and lived over 2,000 years ago. Which brings us to our favorite part. My favorite part is ancient Rome, and anytime I get to talk about Rome, especially ancient Rome, I will do it. And here we have an engineering marvel called the Cloaca Maxima, one of the world's earliest sewage systems. It was Rome's ingenious solution to public sanitation. They also had fort latrines and public latrines representing their advanced infrastructure for hygiene and waste disposal. In the forts, the latrines, they would naturally divert a river or a creek to constantly flow between, and they would allow rainfall to flow and clean out the latrines, and so it would all flow downhill and away from the fort. And with that in mind, they also did it onto a tiny river in Rome. And they first lined this river with cement, and all the sewage would run off into it, and then eventually they covered it and built on top of it. And this ended up being called the Cloaca Maxima. Sorry about that. This ended up being called the Cloaca Maxima, and this investment in public health infrastructure is a significant reason why Rome did not suffer major health outbreaks and why they were relatively rare. They also contributed to a miasma theory. And the miasma theory states that if you would go into these swamps or where the sewage rolled downhill, that you would easily catch something. And that's not too far from the truth. Alright, and so now with the last part of this first video, we're going to move into binomial nomenclature and taxonomy. So let's journey into the systematic world of binomial nomenclature and taxonomy, and you might be surprised to know that the structure and clarity we have today didn't happen overnight. In fact, it took over 200 years, as we'll see. So enter Carl Linnaeus, or Carolus Linnaeus. He is Swedish. This is the Swedish flag. He is a Swedish botanist. Initially, he penned just an 11-page booklet, yes, just 11 pages on Linnaean taxonomy. These pages became the foundational script for what we know today. Linnaeus was a man on a quest, not for treasure or new lands, but to classify and name the vast biodiversity he saw around him. He kind of limited himself to plants and animals, but in his lifetime that was enough. This systematic approach gave to what we know as binomial nomenclature. Okay, and if you just break that name down, it's just bi, two, nomial, names, so two names, and then nomenclature is a system of naming, nomen meaning name in Roman, clature meaning system, so we have a two-name naming system. Okay, so whenever you hear binomial nomenclature, just think two-name naming system. Okay, building on Linnaeus's, in it we write genus, and species in italics. And this is mostly for the written text. Um, in the written text, we go genus, we go species in italics. The genus is uppercase, the species is lowercase. The examples are kind of hidden with the text right here, but it's Bacillus cereus, Escherichia coli, Pseudomonas aerugicina, and a good one is Homo sapien. Homo sapiens. Okay, where Homo is capitalized and it's our genus, meaning man, and sapiens being our species, meaning wise. And so whoever named us called us the wise men or the wise man. Um, and it shows what our early classifiers considered us. Okay. And again, it's hiding it through the text, so this is a very good time to make sure you have looked over any of the um, PowerPoints before you watch these videos. Okay, so then 100 years later comes Ernst Haeckel. Haeckel wrote another probably short essay proposing 4 kingdoms instead of Linnaeus's 2, plants and animals. He added Monera being the bacteria and Proteus being the single-celled organisms that we saw in our pond water. He came in with his iconic tree of life imagery. Okay, he expanded on our understanding by visualizing the connections between these life forms, by making it so that we can relate it to a tree. He really drove it home and kind of influenced how we do modern phylogeny today. 100 years later, Robert Whitaker chimes in with his 5 kingdom model and broadening our understanding of life's diversity, where he adds our fungus. Okay, and it took us 200 years to formulate our 5 kingdom system that we know today, which leads us to our current age. Our current age, we delve into the intricate world of phylogeny. Okay, and if you were to break up phylogeny, it just means the start of branching, right? The branching start. Or the branching type. Modern techniques, particular DNA analysis, allow us to understand evolutionary relationships in unprecedented detail. So with this, we see that the bacteria and archaea split very long time ago, okay, with the distance of this graph being time, or evolutionary time, if you will. So the bacteria and archaea split a long time ago. Archaea have now had a long time without any eukaryotic influence, and they've kind of evolved separately from bacteria. When the eukaryote show up, it takes a while to get to multicellular organisms, all the way to here. It takes that much more time, and in which case we break off into animals, plants, and fungi. And here we are at the animals, and which is why we are called Homo sapiens. Okay, so in essence, Linnaeus's 11 pages today have influenced today's expansive genetic databases. We see an ever-evolving story of life's intricate tapestry in our quest to understand it through microbiology. All right, and with that, we'll stop this lecture. You can review it at any time you want, and I will go into the list of microorganisms on our next video.